Welcome to the Absorption Systems tutorial on the Biopharmaceutics Classification System. I'm Patrick Dentinger, President and CEO of Absorption Systems. My goal for this video is to explain in detail the Absorption Systems approach to performing in vitro permeability studies in support of FDA-approved biowaivers. A biowaiver application has three components: dissolution data, solubility data, and permeability data. Dissolution and solubility assays provide physical-chemical data that are easily interpreted as absolute measurements. FDA requirements for solubility and dissolution measurements are clearly defined in the BCS guidance. Permeability measurements, in contrast, provide rank order data. This means that it's not the absolute value of permeability that is important, but rather how this permeability value compares to that of known high and low permeability reference standards run in the same assay. Appreciating the fact that not all FDA reviewers are experts in the field of in vitro permeability assessment, we have prepared this technical presentation to help you understand our in vitro permeability test system. In addition, we have compiled a list of the most frequently asked questions by FDA reviewers and inspectors over the years. Our detailed responses to these questions can be found toward the end of the video. As you watch the video, feel free to jump ahead or jump back anytime you choose. Let's get started.
The BCS officially became part of the U.S. drug regulatory process in the year 2000 with the release of an FDA guidance on the subject. In 2001, Absorption Systems performed its first BCS assessment, and the data was included in a biowaiver application that was submitted to the FDA by the sponsor of generic fluoxetine. In fact, that study resulted in the first ever BCS biowaiver approval. Shortly thereafter, our BCS classification protocols were audited by the agency, and we were audited again in December 2007 and July 2008. As awareness and acceptance of the BCS grew, the number of requests that we received for BCS studies increased steadily. By the end of 2007, we had performed a total of more than 50 such studies, about half of them in that year alone. Now let's look at the BCS process. The BCS categorizes drugs based on their solubility and permeability. As you know, the criteria for biowaiver consideration require that an active pharmaceutical ingredient, or API, be in an immediate release solid oral dosage form and not have a narrow therapeutic range. The sponsor is responsible for providing data on the dissolution rate of the formulation, stability in gastric and intestinal fluids, and the therapeutic index of the drug. Absorption Systems focuses on two intrinsic properties of the API: permeability and solubility. Most of this video will focus on permeability. For any highly soluble and highly permeable APIs, Class 1 compounds, scientific evidence suggests that the fraction of the dose absorbed will be high and consistent from patient to patient for comparable formulations. This is the rationale for the biowaiver provision of the BCS guidance. When a biowaiver is granted, a clinical bioequivalence study can be replaced by in vitro testing. In addition to Class 1 compounds, there are a few recent examples of biowaivers being granted for Class 2 and Class 3 compounds. A biowaiver can simplify the clinical development of a drug. That's why the FDA formalized the process. It's part of the Critical Path Initiative to speed up drug development. In addition, an in vitro BCS classification is definitive and precise, unlike a clinical study. In a clinical study, a drug may have low oral bioavailability, or BA, and you don't know if it is poorly permeable or highly permeable but rapidly metabolized. With regard to solubility, it's very straightforward, and we follow the BCS guidance verbatim. An API is considered highly soluble if the highest dose strength is soluble in 250 milliliters or less of aqueous medium over the pH range 1 to 7.5. Permeability is much more complicated than solubility because it involves developing a set of reference compounds to validate the in vitro permeability model, choosing a suitable model, optimizing the model, validating the model, evaluating model validation data, selecting a high permeability reference compound, and dealing with unanticipated behavior of APIs in the validated test system, namely, low recovery caused by cellular accumulation or nonspecific binding to the testing apparatus, multiple sampling schedules caused by permeabilities that differ by orders of magnitude, and cytotoxicity at high concentrations of API. The asterisk indicates that our validated system includes a significant advance in this respect, which we will highlight shortly. The permeability class boundary is based on the fraction absorbed in humans. As long as an API is stable at gastric pH, it is considered to be highly permeable when its permeability in the test system is higher than that of a co-dosed referenced compound whose fractional absorption in humans is known to be at least 90%. The in vitro model that we use to assess intestinal permeability is the CACO2 cell monolayer. CACO2 cells are polarized human intestinal epithelial cells, meaning that they form a single layer of interconnected cells with different properties on their apical and basolateral surfaces. In the body, the apical surface of an intestinal epithelial cell is the surface that is exposed to the lumen of the small intestine, and the basolateral surface is exposed to the blood. Before performing a BCS study to support a request for a biowaiver, we had to validate the relationship between in vitro permeability in our test system and the known in vivo fractional absorption of a series of more than 20 compounds. The validation ensures that our test system will produce reliable and reproducible results to support regulatory submissions. Our validation report is available in a secure area of our website, and if you are watching this video, you may already have access to the report. Here we are correlating the apparent permeability, or P apparent, across CACO2 cell monolayers in the apical to basolateral, or A to B direction, with the fraction of dose absorbed for a series of orally administered drugs. As the plot demonstrates, P apparent values above a certain threshold are always associated with high fractional absorption. Note that the relationship between the two parameters is not linear, it is sigmoidal. In addition, the clinical data on the y-axis is taken from the literature, and in some cases the reported values for fraction absorbed can vary significantly. For example, gliclazide is a drug that has a high in vitro P apparent value and reported values of fraction absorbed ranging from 65% to greater than 100%. In spite of this degree of uncertainty, a suitable reference standard is able to categorize an unknown API as high or low in a properly validated test system. 90% oral absorption is set as the high permeability threshold in the BCS guidance. Therefore, our validated in vitro test system includes a reference compound that has at least 90% oral absorption. We co-dose each API with the reference compound, and if its P apparent is at least as great as that of the reference compound, the API can be categorized as highly permeable. This graph illustrates the power of our validated CACO2 cell monolayer system to distinguish between compounds that are well absorbed and compounds that are poorly absorbed in humans in vivo. On the y-axis is the in vitro P apparent in our system. There is about a 5-fold difference in P apparent between the most permeable compound in the low permeability class and the least permeable compound in the high permeability class. This large dynamic range permits a sharp discrimination between BCS class 3 and 4 compounds with low permeability and class 1 and 2 compounds whose intrinsic intestinal permeability is high. In 2006, we optimized our existing validated system by switching from pindolol to minoxidil as the high permeability reference compound. One advantage of the switch has to do with the pH at which in vitro BCS studies are run. The BCS guidance doesn't tell us what pH to use. We always keep the pH of the basolateral buffer at 7.4 because that is consistent with the physiology of intestinal epithelial cells in the body. But in the body, the apical pH can vary from quite acidic in the duodenum to pH 7.4 or even higher in the ileum or the colon. Pindolol, our old reference compound, was very sensitive to apical pH in our in vitro system. But for minoxidil, the new reference compound, there is no difference in P apparent at pH 7.4 versus 6.5. FDA inspectors have told us that they like the fact that the new reference compound behaves the same way at either pH. As we said, the CAECO-2 assay system categorizes permeabilities on a rank order basis. That is, the value of P apparent of an API must always be compared with that of a co-dosed reference standard in order to properly categorize the permeability of the API. It's still useful to see how variable the absolute P apparent values are when they are measured under carefully controlled assay conditions. In this table, we summarize A to B and B to A permeability values obtained for pindolol. In any given experiment, we can expect the A to B P apparent to range from about 7 to 19, a range of 2 standard deviations above and below the mean. The permeability assay system includes CAECO-2 cell monolayers grown in transwell devices. Transwells consist of 2 parts, an insert and a well. The bottom of the insert is a semi-permeable filter, on which a layer of CAECO-2 cells 1 cell thick, a monolayer, is cultured. To establish a monolayer culture on the filter, CAECO-2 cells mixed with culture medium are added to an empty insert. The cells settle to the bottom of the insert, attach to the collagen-coated polycarbonate filter, and begin multiplying. The culture medium is replaced in both chambers every couple of days. When the CAECO-2 cells fully cover the filter, they have reached a stage termed confluence, and the cells begin to differentiate and polarize. The apical surface of the cells is exposed to the upper chamber, and the basolateral surface faces the filter, which is in contact with the medium in the lower chamber. By about 21 days in culture, the monolayer of CAECO-2 cells has fully differentiated and expresses a phenotype, characteristic of intestinal cells. There are tight junctions between them, and the cells express uptake and efflux transporters commonly found in the human small intestine. The monolayer now behaves as an in vitro piece of human intestine. It is this property that makes the CAECO-2 cell monolayer a preferred in vitro model for studying human intestinal transport of drugs, nutrients, toxins, herbal extracts, anything that is ingested orally. Some compounds cross the monolayer primarily through the tight junctions between cells, also called the paracellular route. Because tight junctions occupy only a few % of the total surface area of the monolayer, such compounds have low P apparent values. Atenolol and lucifer yellow are examples of such compounds. Any breach of monolayer integrity leads to a dramatic increase in the permeability of these compounds, which is why they are used as controls in the CAECO-2 assay. Highly permeable compounds, in contrast, move primarily through the cells. During a permeability assay, the culture medium is replaced by a buffered isotonic salt solution containing glucose to provide a simpler matrix for bioanalysis. The basolateral pH is always maintained at 7.4. The apical pH, however, can vary somewhat to mimic the different pH environments found in different regions of the small intestine. We measure the permeability of drugs in both directions. The permeability in the apical to basolateral, or A to B direction, is measured by adding drug to the upper chamber and measuring the amount that appears over time in the lower chamber. B to A permeability is measured in the same way, but in the opposite direction. We sample from the donor side, which is the A side in an A to B experiment, at the beginning and end of the study. We sample from the receiver side, which is the B side, at several time points to ensure we are in the linear portion of the time course. Each time we sample from the receiver side, we replace with fresh buffer to maintain the so-called sink condition, which is an implicit assumption in the calculation of P apparent. One thing that can confound the results of an in vitro permeability study is nonspecific binding to the transwell device. This is reflected in low recovery of the API, which can lead to a false negative, that is, a highly permeable compound being mistakenly classified as having low permeability. The high permeability reference compound minoxidil, as well as the monolayer integrity markers atenolol and lucifer yellow, are well behaved and don't show any evidence of loss to the device. Compounds that bind nonspecifically to polystyrene or polycarbonate or accumulate in the cell monolayer will have low recoveries. For this reason, the first thing we do after receiving an API is a preliminary BCS study. This serves as a system suitability study to determine whether or not the API is compatible with the CACO2 monolayer assay system and to help develop the pivotal BCS study protocol. One of the purposes of the preliminary study is to determine the recovery of the API in a cell-free transwell apparatus. If the recovery is low, we can often mitigate the problem by pre-incubating the device with the API to occupy most of the nonspecific binding sites. Testing for low recovery ahead of time helps us select the optimal conditions for the pivotal study to ensure that we can interpret the results properly. We also determine recovery with a monolayer in the device during each permeability test. For well-behaved compounds, the sum of the amount remaining in the donor well at the end of the experiment plus the total amount that crossed the cell monolayer into the receiver well is equal to the amount that was added at the start of the experiment. Typically, recoveries of at least 80% are considered complete. If pre-incubation of the device does not improve recovery to at least 80%, we extract the cell monolayer with an organic solvent to determine the residual concentration associated with the cells. The guidance tells us, at the end of an in vitro test, we need to determine the amount of API associated with the monolayer. For BCS classification, we need to know the A to B permeability of the API across the CACO2 monolayer. But in the preliminary study, we measure the permeability in both directions to find out if the API is a substrate of an efflux transporter. Drugs that are substrates of apical efflux transporters, such as P-glycoprotein, have higher permeability in the B to A direction than in the A to B direction. This is because efflux transporters help the drug to get out of the cell in the B to A direction and keep the drug from getting into the cell in the A to B direction. The parameter that we look at is the ratio of the two, called the efflux ratio. Non-substrates have an efflux ratio of 1 because they don't interact with an efflux transporter. A drug that is a substrate of an efflux transporter, on the other hand, would have a high efflux ratio. If it is an efflux substrate, our in vitro test system might underestimate its permeability, and the guidance says that in that case, bioavailability has to be determined in a clinical study. At this stage, we also determine tolerability. In other words, can we test the recommended concentration without damaging the cell monolayer? Now we will cover one of the most challenging aspects of validating our in vitro permeability system, selecting the most appropriate time points for the study. This will hopefully help explain the rationale that we use and have validated for selecting time points in a BCS permeability study. We typically see 3 different patterns of compound accumulation in the receiver compartment during a permeability test. The red line represents a highly permeable compound, which shows a rapid initial rate of increase followed by a leveling off as the concentrations in the donor and receiver compartments tend toward equilibrium. The initial rate is the one that we use to calculate the P apparent value. Only highly permeable compounds reach a plateau within the duration of a CACO2 experiment, which is typically 2 hours. The green and yellow lines also represent compounds with high permeability, but not so high that the rate of accumulation plateaus within a 2-hour experiment. Finally, the blue line represents a low permeability compound, such as atenolol, the control compound we co-dose with an API to confirm the integrity of each of the monolayers used in a permeability study. The plateauing of the high permeability compound and the very different rates of accumulation for the high and low permeability compounds mean 2 things. First, we have to be very careful which time points to use for calculating the rate of accumulation of the high permeability compound. And second, we have to use different time points for the low permeability compound, because there isn't enough of it to analyze at the early time points that we have to use for the high compound. The low reference compound is easy. It's something that we use over and over. The recovery is high, and we know from experience that its accumulation in the receiver compartment is linear for at least 2 hours. Figuring out which time points to use for a high permeability compound, particularly if it is an API that we don't have a lot of experience with, is more difficult, as we will now illustrate. In this case, If we picked the 30-minute time point and forced the line to go through the origin, we would slightly underestimate the true rate of accumulation, which is linear between 10 and 30 minutes. The underestimation is much more severe if we use a single longer time point, such as the 90-minute point illustrated here, and assume that the rate is linear from time zero. For this set of data, the appropriate time interval to use in calculating the rate of accumulation is 10 to 30 minutes. Our BCS study protocol specifies that we use the first 3 quantifiable time points, which means that we never force the line to go through the origin. What if we spaced out the sampling time points a little more, say 15, 30, and 45 minutes, so that accumulation of the API started to plateau before the 3rd quantifiable time point? In that case, we would underestimate P apparent to some degree. Here is an even more difficult case to analyze. A highly permeable compound with a lag before it appears in the receiver compartment, typically caused by intracellular accumulation of the compound. Very hydrophobic compounds frequently show this type of behavior. Such compounds also tend to bind nonspecifically to the apparatus. Pre-incubation with the API helps alleviate the lag as well as the low recovery. Also, the concentration in the donor compartment at the end of the lag period can be quite different from the initial dosing concentration because a lot of the compound is inside the cells by that time. So what concentration do we use as C0 in the P apparent equation? Although the actual concentration at time zero may be lower than nominal, our protocol is to use the nominal concentration. Including t equals 0 in the slope calculation severely underestimates the true P apparent value. But as we already said, we never force the line to go through the origin. The true initial rate of accumulation in this example would be determined from the linear portion of the curve between about 30 and 50 minutes. But our protocol is to use the first 3 quantifiable time points. In this particular example, we would use the 30, 60, and 90-minute time points and calculate a rate that would slightly underestimate the permeability. The next step is to get an idea of the solubility of the API. Based on the results of the preliminary solubility and permeability tests, we get an idea of the API's BCS classification. Then we consult with a sponsor before finally moving on to the definitive BCS classification study. This is a very rigorous study that includes full GLP analytical methods validation, as well as solubility and permeability assessment exactly as specified in the BCS guidance.
So that's the Absorption Systems BCS story. We hope this presentation has helped you understand our approach to in vitro permeability measurements in support of BCS biowaivers. Remember, you can go back to any section that you want to see again. Feel free to contact us with any additional questions. Thank you very much for your time.